My name is Brandy Likes, and I'm the Chemistry Section Supervisor for the St. Louis County Police Crime Laboratory. Today I'm going to introduce you to forensic chemistry and take you on a tour of our section. The chemistry section is made up of 3 distinct disciplines: seized drugs, fire debris, and explosives. Our chemists analyze evidence from over 60 different police departments within St. Louis County and federal agencies, including ATF, DEA, and FBI. Before we get started on our tour, I would like to talk briefly about what it takes to become a forensic chemist, as I get asked this question often. While it is a requirement that you have a degree in a natural science, it is not a requirement that you have a chemistry degree. That said, we are a working analytical chemistry lab, so certain courses are required, such as Organic Chemistry 1 and 2 and Quantitative Analysis or its equivalent. We need scientists with a basic understanding of the fundamentals of chemistry. We'll teach you how to become a forensic chemist. So we're going to begin our tour in the drug section, where chemists analyze unknown powders, liquids, tablets, and plant material for the presence of controlled or illicit substances. Examples of commonly seen illicit substances that we see in our crime laboratory include cocaine, fentanyl, and methamphetamine. When we refer to things as being illicit or controlled, what we're talking about are drugs that are listed either federally or on the state level as being in Schedules 1 through 5. For example, Schedule 1 drugs such as heroin have no known medicinal uses and have a very high potential for abuse. Schedule 2 drugs such as fentanyl also have a high potential for abuse but have known medicinal uses. Our chemists must also stay informed of new drugs entering our region. This includes ensuring that our analytical techniques are current, that we have known standards available, and that we stay informed of current legal status. We routinely have new drugs enter our region that have the potential to be very dangerous, but they aren't currently controlled. Our chemists use a variety of analytical techniques and instrumentation to include color test, FTIR, and GCMS. Our chemists also spend a great deal of time educating law enforcement on things such as drug trends, drug labs and precursors, safe handling, and proper packaging. So now we're going to demonstrate a color test. A color test is just a simple presumptive test that provides us with an indicator chemically of what we're dealing with. Now, that said, color tests do not confirm the presence of controlled substances. What they do is sort of lead us in the right direction of maybe a specific drug class or a specific chemical structure. The color test that we're going to demonstrate today is a 2-part color test known as Simon's test. And what Simon's test does is it reacts in the presence of a specific chemical structure called secondary amines. Examples of drugs that are secondary amines include methamphetamine and MDMA. When we perform a color test, we typically add the reagent to an empty spot well to ensure that it's clean. As you can see, there's no reaction. We then add a small amount of the sample and wait for a reaction. As you can see, we have a nice blue color reaction. Again, this is an indicator of a possible secondary amine. This does not confirm the presence of a controlled substance. So now that we've performed our color test and got an indication that we have a secondary amine, we must now confirm the identity of the controlled substance. One of the instruments we use for confirmation is an FTIR. We simply take a small portion of the sample and place it on the sample window. We then crush the sample to ensure sample distribution on the sample window. So the spectrum produced now is unique to the molecular structure of our unknown compound. It's basically a molecular fingerprint of that structure. So now we are entering the main chemistry instrument lab. As you can see, this laboratory is full of instrumentation to include mostly GC mass specs. So GC mass specs are another instrument we use for the confirmation and identification of controlled substances. Unlike the FTIR, the GC mass spec actually allows for separation and identification of every component in an unknown mixture. Once we've reviewed all of our analytical data and completed our case file, a final report may be issued. Now we're going to tour the fire debris and explosives laboratory. On the fire debris side, chemists analyze samples for the presence of ignitable liquids. Ignitable liquids include things such as gasoline, charcoal lighter fluid, and tiki torch fuel, and are used to accelerate a fire. Determining the presence of ignitable liquids assists fire investigators determine origin and cause in suspicious fires. Now, our cases are not limited to only commercial and residential fires, but also include homicides, insurance fraud, serial arsonist, civil unrest, and incendiary devices such as Molotov cocktails. On the analysis side, the same GC mass spec that's used to confirm and identify the presence of drugs is also used to confirm the presence of ignitable liquids. That said, sample prep for fire debris is very different. Packaging is very important for fire debris samples, as the whole goal of our analysis is to try and collect any ignitable liquid vapors that may be trapped within the sample. We want to make sure that we have plenty of room in the evidence can for these vapors to move around as they're heating. So Ryan is actually going to take the activated charcoal strip and he's going to hang it on the inside of the can. And he's going to do this by placing a magnet on the outside of the can. He's then going to take a paper clip and insert the activated charcoal strip and simply hang it on the inside of the can. He will then reseal the can and we'll place it in one of the 2 ovens that we have. We heat these samples in order to increase the mobility of the ignitable liquid vapors that may be present in order to adhere to the activated charcoal strip. Chemists on the explosive side provide support to the St. Louis Regional Bomb and Arson Unit. This includes bomb squads from St. Louis City Police, St. Louis County Police Departments, St. Charles County, and federal agencies to include FBI and ATF. Explosives evidence may only be submitted by certified bomb technicians or certified explosive specialists. Explosives analysis involves the identification of explosives, explosive precursors, chemicals found at homemade explosives labs, and components used to assemble improvised explosive devices such as pipe bombs. Chemists use a wide variety of analytical techniques to include GC mass spec, FTIR, X-ray diffraction, and SEM. However, before the analysis can begin, the chemist must first document all the components submitted. This includes taking measurements, taking photographs, and taking the individual components and looking at them under a stereoscope, looking for particles and residues that may be used for further analysis. This is the Scanning Electron Microscope, or SEM. The SEM is a very powerful tool in our laboratory and it's used for the analysis of explosives. The SEM is a microscope, however it's a very powerful microscope that allows us to image tiny particles such as post-blast particles. Our SEM also has an X-ray detector. This detector allows us to not only image these tiny particles, but we can determine the elemental makeup of those particles. Our chemists may be called to assist regional bomb squads in the field by providing guidance on explosives, explosive precursors, chemical hazards, safe handling, and proper packaging. Our chemists also receive ongoing training on explosive trends that are occurring both domestically and globally. We use this information to then teach local bomb squads and military on the awareness and recognition of homemade explosives. I hope y'all enjoyed that little insight into forensic chemistry, and thank you for stopping by.
Hi, my name is Tina Wilson. I am a firearm and toolmark examiner for the St. Louis County Police Crime Laboratory. We are the last section that evidence travels throughout the lab, so if it needs to go to biology or fingerprints for processing, it will travel to those sections before it comes to us. We are a pretty low-tech section for the laboratory, so we typically use a microscope and do most of our analysis at our lab bench. Come into the lab with us. Let's go take a peek. Here at St. Louis County, we perform function testing of firearms that are seized off of the street and submitted to the crime laboratory. We perform microscopic comparisons of fired cartridge cases and bullets. Those can be found at a scene or those can be from test shots from a firearm. We also perform serial number restorations where we restore serial numbers that have been obliterated on a firearm, and we perform toolmark comparison analysis as well. Toolmark examination is the comparison of characteristics left behind when one object hits another object. The harder object strikes the softer object, leaving behind striated or impressed tool marks. A gun is considered a specialized tool in that aspect. When a firearm is manufactured, the machining process leaves behind unique characteristics on that firearm. Those unique characteristics become even more unique over time through use and wear of that particular firearm. In front of me here, I have a Glock firearm. This firearm, as it travels through the cycle of operation, it is going to leave unique characteristics from this firearm onto a bullet in a cartridge case. Those marks are going to come from the extractor, the ejector, the breech face, chamber marks, as well as from the barrel itself. These marks are all identifiable from that cartridge case and that bullet back to this particular firearm. So this is a Glock firearm. The markings left from the breech face, the ejector, as well as the extractor, the chamber, and the barrel all leave unique characteristics on a cartridge and a bullet when they are fired through this particular firearm. When a firearm comes into the lab, our examination process begins with first photographing a firearm and the contents of the packaging, and then making sure that the firearm is safe. So we do that by ensuring that the magazine is out of the firearm, that there are no rounds in the chamber, that the magazine well is clear, and that there are no items in the barrel. We are the only people in the world who probably look down the barrel of a firearm, but we always stick something in the chamber before we do so. That way, in case there was something in there, it is not going to be capable of firing. Once we have done that, we will function test the firearm. So that is going through basically the cycle of operation here at our desk. So does the trigger safety on this firearm appear to function? If there were other safeties, we would check those safeties. And then we pull that trigger and make sure that it functions properly at our desk. After that, we will take it into our range and we will test fire the weapon. Fire cartridge cases and bullets are submitted to the laboratory. These are typically submitted from a crime scene. We can have multiple cartridge cases and bullets. We can have 1 or 2 cartridge cases and bullets. The first thing that we do with these is we will open them up at our lab bench. We will look at them under our stereoscope here to determine if first they have similar class characteristics. If we determine that they have the same or similar class characteristics, the next step then is to transition into our comparison microscope, which allows us to view 2 items simultaneously at one time. And we can then further look at these 2 items on the microscope to determine if they have the same or different individual characteristics, which will then tell us if they were fired from the same firearm or from 2 different firearms. I have worked thousands of cases, and in my experience, no 2 firearms have marked the same. After we have test fired a firearm, we are going to bring it into the range to acquire test shots from that particular firearm. So the first thing that I'm going to do is I'm going to grab a couple of cartridges here, load those into my magazine, and now this firearm is ready for test firing. Let's go into the range and get some test shots. So we are able to test fire these firearms on the 4th floor of this building thanks to this range. So this range has bulletproof walls. It is surrounded by thick layers of concrete as well. It is also designed in a way that the end down here is designed like a snail shell. So if you think about a snail and what their shell looks like, how it's kind of circular, that kills the momentum of a bullet and allows us to be able to shoot in this range safely, that it doesn't affect anybody that might be on the other side of that wall. Here we are in our water tank room. So when evidence is submitted and they are requesting comparison with fired bullets found at a crime scene or from autopsy, we need to acquire pristine bullets from a particular firearm. To do so, we will fire those test shots then in this water tank here, and that will give us that pristine bullet for comparison. So let's take some test shots. Now that we have our test fired cartridge cases and our test fired bullets, we are ready to go into the microscope and perform our microscopic comparison. The NIBIN database, which stands for the National Integrated Ballistic Information System, is a database of 3D images of fired cartridge cases from scenes and test shots from firearms that are submitted to the laboratory. Those 3D images then come back to us so that we can correlate against different crime scenes to determine if cartridge cases from one scene are related to a gun that later came into the lab, or vice versa, if a gun that we get into the lab is related to any previous scenes. This is our firearm reference collection. Our reference collection is made up of hundreds of different crime firearms. These crime firearms are used for training purposes, not only for new examiners, but also for recruits, crime scene classes as well. We also use these firearms in the event that a gun comes in and it does not function, and we need a part to make that firearm function. We can pull that part from this gun. We can also use these firearms as a reference for serial number restorations for the serial number format, as well as just for having some fun. Those interested in pursuing a career in firearm and tool mark analysis will need to have a bachelor's or advanced degree in a natural science or a forensic science field. Once hired into this section, you will then undergo a 2-year training program as outlined by the Association of Firearm and Tool Mark Examiners. Within that 2-year training program, you will attend manufacturing facilities, you will attend armors courses, you will perform numerous in-house practicals and training assignments, as well as work supervised casework under the direction of a trainer. It is rewarding to know that the work that we are performing here in the firearms section of the St. Louis County Police Crime Laboratory has an impact on not only the residents that we service, but also on surrounding areas. It is just as important to determine that a fired cartridge case or a fired bullet was not fired from a firearm to let investigators know what direction to take on a possible investigation.
Welcome to the biology unit of the St. Louis County Police Department. In the biology unit, we are internationally accredited, and we also work cases such as property crimes and crimes against persons. But wait, before we come into the lab, you have to wear personal protective equipment. We always wear a lab coat, a face mask, and gloves. For purposes of the video today, I will not be wearing a face mask. Anyone coming into the lab must submit a DNA sample for elimination purposes. The biology DNA unit is one section. The biologist handles screening the evidence for blood, semen, saliva. We also get evidence with trace samples such as sweat that is left behind and skin cells. In addition to screening for biological fluids, the biology section is also responsible for triaging the evidence. Not all evidence collected at a crime scene may be relevant to the crime. The biologists decide what items should be tested and which body fluid. A portion is then placed into a tube, which will then go through the DNA process. You will learn more about that soon. The cases in biology take a few hours to a few days to complete, depending on how much evidence is submitted. Despite what you see on CSI, this all takes time. After the biologist finishes the work in the actual laboratory, a biology report is written, and if the case included evidence that needed to be tested for body fluids, then that report must be reviewed by another biologist for quality control. The biologist also may be needed to testify in court about their findings. Let's take a look at some of the tests the biologists perform to identify bodily fluids. For example, this piece of carpet, you can't see anything with the naked eye, but when we use the alternate light source, as you see, the stain becomes visible. After the alternate light source, the swabbing of the stain is then tested with STMP reagent. A blue color change indicates a presumptive positive result for seminal fluid. When screening for blood, the IR light is a tool used to locate blood on dark or colored objects. The phenolphthalein test is a sensitive but not specific test used to detect hemoglobin in blood. Another commonly used reagent to detect blood is luminol. Luminol is used as an investigative aid to detect blood that is not visible with the naked eye. It is commonly used to detect blood at crime scenes that may have been cleaned up. The amylase diffusion test is a presumptive test used to indicate whether an elevated level of amylase exists in an unknown stain or swab. If so, this may indicate the presence of saliva. To perform this test, a small sample is extracted in sterile water for approximately 45 to 60 minutes. Small holes are then punched in a gel plate using a pipette to produce a well, which is approximately 2 millimeters in diameter. The samples are then placed in the wells using a pipette. Each well holds approximately 4 microliters of liquid. The Petri dish is then placed inverted in an incubator at approximately 37 degrees Celsius for 2 to 6 hours, or it can be stored at room temperature overnight. The plate is then stained by pouring a dilution of saturated iodine solution over the gel surface and then rinsed with water. A clearing around a well indicates areas of amylase activity. The diameter of the clear circle is proportional to the square root of the concentration of amylase. An approximate measurement of the diameter of each ring present is then taken and noted. A positive sample is when the ring size is equal or greater in size than the positive control. If the ring is less than the positive control, but greater than the negative control, it is inconclusive. A negative result would be the absence of any clear ring. The heme-direct test works similar to a COVID test and is a confirmatory test for blood of probable human origin. It is not considered a confirmatory test for species due to the fact that some upper primates and under some conditions, ferrets have been reported giving positive results with the heme-direct card. This test is performed by cutting a very small portion of the sample to be tested. The sample is then added to the extraction buffer. The sample is vortexed and sits at room temperature for approximately 10 minutes. The tip is then snapped off of the top and several drops from the dropper of the extracted sample are added to the sample well of the testing device. We wait 10 minutes to read the results. A pink line in the control and test column indicate a positive result. After the items have been screened and sampled, they are placed into these tubes. These tubes are then placed in the freezer for the DNA analysis.
Hi, my name is Justin Lautz. I'm a forensic DNA analyst with the St. Louis County Police Department Crime Laboratory. And we're going to do a brief tour of the lab and the things that we do in the DNA analysis unit. So come along, let's take a quick tour. But before we go into the lab, we have to put on personal protective equipment. And that is going to protect us from the evidence and the evidence from us. Normally I would wear a face mask, but we're going to take special considerations today where I will not have to wear a face mask. Our primary responsibilities in the DNA analysis unit is to take items of evidence that have been previously analyzed in our biology unit and perform DNA analyses on them. For this position, there are specific educational requirements. You must have a bachelor's degree in a biology, chemistry, or forensic-related area. The degree must include coursework, graduate or undergraduate level, in biochemistry, genetics, molecular biology, and statistics or population genetics. In addition, extensive on-the-job training is required and typically takes anywhere from 1 to 2 years to complete. So what is DNA? DNA stands for deoxyribonucleic acid and is contained in every nucleated cell in your body. Half of it comes from your mother and half of it comes from your father. It's essentially your genetic blueprint. It dictates your chemical composition as well as your physical appearance. The first step in this process is extraction. This is where we are going to clean the sample as well as get DNA into solution, or what we call the lysate. Here we have an analyst adding reagents in the first step of the extraction process. We need to remove the proteins, cell membranes, and other parts of the sample that we're not interested in. This step is called extraction because we're extracting the DNA from the sample. In our laboratory, robotics do the final extraction step in our process. This is performed on the QIAGEN EZ1 as well as the Tecan Evo Freedom 100 liquid automation instrument. Once the robots are completed with the extraction process, the sample is then eluted in about 50 microliters of sample, about the equivalent of a raindrop. The next step of the process actually takes place in a different room in the laboratory. This is known as the pre-amplification room. And this next step we're going to embark on is quantitation. And quantitation is exactly that. It is determining how much DNA was in the sample that we just recovered during the extraction step. Some samples contain large amounts of DNA, while others contain only a few cells and may not have enough DNA to get a usable result. For the quantitation assay, a tiny portion of the 50 microliter DNA extract is taken and placed into a PCR plate along with special reagents. We use real-time PCR instruments to perform the quantitation step. As the quantitation reagents copy certain areas of the DNA molecules in the sample, a fluorescent signal is emitted. The relative brightness of the signal is measured by the instrument and is converted into an estimate of how much DNA is present in each sample. The third step in the process is amplification. If you could think of your genome as a book, there's only certain pages of that book that we're interested in to make a forensic identification. This process is in part thanks to polymerase chain reaction. During the PCR process, fluorescent tags are added to the DNA fragments as they are being copied. A specific volume of the DNA extract is added to a PCR plate containing the amplification reagents. The volume of DNA extract used depends upon the quantitation results. The instrument used to perform the PCR process is called a thermocycler. It contains an extremely precise heat block that raises and lowers the temperature of the samples at specific times. During this process, the DNA is heated to near boiling temperatures. The last step of the process is to detect the sample fragments that were amplified and fluorescently tagged. This process is known as capillary electrophoresis, or CE. An instrument called a genetic analyzer is used to separate the DNA fragments by size as the fragments pass through the capillary. The smaller fragments move more quickly and are detected before the larger fragments near the end of the capillary. A laser excites the fluorescent tags, causing them to emit light. This light is captured by a highly specialized CCD camera that collects the light emission data. This data is converted into a DNA profile by genotyping software. Once the samples have finished processing on the CE instrument, the data is analyzed in a program called FASTER. This software takes the light emission data from the genetic analyzer and converts it into DNA data that the analyst can interpret. The DNA analyst evaluates the data to determine if sufficient data was developed from the samples. If enough data was recovered, they determine the number of contributors in the DNA sample. DNA samples often contain DNA from more than one individual. This is called a mixture. Some mixtures are simple, while others are more complex. Depending on how many individuals are present and how much DNA each person contributed, it may not be possible to determine if a specific person is present in a mixture. Other DNA samples have no DNA at all, or only a few peaks that are not enough to be useful. Samples that are able to be interpreted can be compared to known DNA profiles from individuals in the case. If associations or matches are considered relevant in the context of the case, statistics are applied to help the jury understand how strong or weak the association is. This is where a software program called STRmix is used. STRmix is a highly validated software that implements the most modern DNA interpretation methods. The software untangles complex DNA mixtures and resolves them into a list of genotype combinations that best explains the DNA profile. These combinations are compared to DNA profiles from individuals in the case to determine the potential source of the crime scene sample. When an association is made and the association is relevant in the context of the case, a statistic called a likelihood ratio is calculated using STRmix. The statistics aid the jury by providing weight to the association. It helps the jury understand if the association is strong or possibly weak. These results are summarized in the DNA report. Prior to release, a second qualified analyst must review all the lab work as well as the results, interpretations, and conclusions contained in the DNA report. In some cases, some samples can be entered into a DNA database where they can be compared upon other evidentiary samples and known standards. This is known as the Combined DNA Indexing System, or CODIS for short. Federal law dictates who has access to the CODIS database and what information is allowed to be entered. DNA data entered in the database contains only the DNA profile and sample identification numbers, but unlike what you see on crime dramas, does not contain any personally identifiable information. When a match is made, the labs involved must correspond to determine whether or not it is a true hit. If determined as a true hit, this can give investigators an investigative lead. Thanks for coming along in this unique opportunity to see the inner workings of our DNA lab. As you can tell, the road to getting a DNA profile is long and nothing like you see on TV.
My name is Katie Cohen, and I'm a certified latent print examiner with the St. Louis County Police Department Crime Lab Fingerprint Unit. Today I'm going to walk you through the fingerprint unit to give you an idea of what we do here. We analyze and compare latent prints found at crime scenes. Latent means hidden, and typically a latent print needs further processing to be seen. Your fingers, palms, and soles of your feet are made up of friction-ridged skin. Your friction-ridged skin, or your prints, don't change over time. The ridges of your prints are made up of sweat pores, so when you touch something, there is the possibility of leaving behind an impression or a copy of your print. We have a variety of processing techniques, both physical and chemical. The type we use depends on the surface type of the item. For non-porous items like soda cans and guns, we can use cyanoacrylate, dye stains, and what you most commonly see on TV, fingerprint powder. For porous items like paper, we can use ninhydrin or indane-dione. We also have chemicals for processing the sticky side of tape and items contaminated with blood. Your prints are unique to you, so no two people have been found to have the same fingerprints. Even identical twins have different fingerprints. That's how we're able to use them as a form of identification. When comparing a latent print to a 10-print card from a known individual, we are looking for the same details in the same spatial arrangement in both prints. So if you enjoy the Spot the Difference picture puzzles and highlights magazines, you might find this type of work enjoyable. The fingerprint technicians are tasked with verifying the identity of those fingerprinted and booked through the system. They do this by comparing the fingerprints obtained from that individual to any other records that we have on file down here. Once arrested, an individual is brought to Justice Services, where they are fingerprinted using our LiveScan device. LiveScan is a tool used to digitally capture an individual's fingerprints and palm prints. Those prints are then sent to both the Missouri State Highway Patrol and the FBI through the Automated Fingerprint Identification System, also known as AFIS. Once uploaded, every individual is assigned a state ID number as well as an FBI number. Once in the AFIS system, these known prints can then be used by latent print examiners when trying to identify unknown prints from crime scenes. By comparing fingerprints, the technicians are able to determine when somebody isn't giving their true identity when arrested and using an alias. This ensures that nobody can use your name when they are arrested. The fingerprint technicians are vital in ensuring that you are who you say you are. Anyone interested in becoming a latent print examiner would need a bachelor's degree in either biology, chemistry, a forensic science related field, or criminal justice. Latent prints takes a lot of patience, training, and practice, and it's very different than what you see on TV. While working as a latent print examiner isn't always a walk in the park, it is great being able to assist law enforcement investigations and give closure to grieving families. Thank you for joining me on this tour today, and I really hope you learned something about what we do here.
Hi, my name is Kristen, and I'm a computer forensic examiner here in the Digital Forensics Unit of the St. Louis County Police Department Crime Lab. Here in digital forensics, we investigate any crime that has a digital element. That can be anything from a computer to a cell phone to a variety of other devices. It's estimated that at least 90% of all crimes have a digital element, and here in our unit, we work over 1,000 pieces of digital evidence per year. So let's head on inside and take a look at what we do. For our first discipline here in the lab, we have surveillance video and router pulls. For these, we often respond to homes and businesses in order to pull video footage or router information that's pertinent to an investigation. Because of this, we're the only civilian unit in the entire police department that has an on-call rotation that's 365 days a year, 24/7. For routers specifically, we connect our computers directly to the router itself in order to extract the data that is pertinent to the investigation. So moving on to mobile device forensics, when we first get a cell phone in our lab, we want to keep it from connecting to the outside cellular network, and we do so through the use of Faraday bags or the Faraday room that we have here in our lab. Our next goal would be to extract the data that's needed for the investigation from the phone itself. And the success of this usually depends on things such as the make and model of the phone, as well as the operating system that it's running. Once we've obtained an extraction from a mobile device, we then take steps to ensure that the copy of the data is accurate. We then move on to our analysis. During analysis, we look for things such as pictures, text messages, location data, and countless other things, kind of dependent on what the case officer has requested us to look for that's pertinent to the investigation. For the computer discipline in our lab, we examine a variety of devices to include desktop computers, laptops, external hard drives, and thumb drives, just to name a few. The next step in the process would be to create a forensic image or a bit-for-bit copy of the data extracted from the device. We then move on to the analysis portion based on the needs of the case officer, which often includes looking for things such as photos, videos, or emails. Historically, many of the computers that we work have been for our Internet Crimes Against Children cases. We directly support our Special Investigations Unit with their Internet Crimes Against Children cases, and we report our findings directly to the National Center for Missing and Exploited Children. Due to the disturbing nature of these cases, mental health is a top priority in our unit, and we try our best to support our examiners in this way. One of the biggest challenges in our unit is keeping up with the rapidly changing technology. For instance, anytime your phone gets an update, our job changes. Thanks for visiting us today. If you'd like to be a computer forensic examiner in our unit, you don't necessarily have to have a college degree, but one in a computer-related field is preferred. In your first 2 years of employment, you'll be required to obtain the Computer Forensic Certified Examiner's certification, as well as undergo a variety of hands-on, on-the-job training. Overall, this is a growing and exciting field, and it's definitely the future of criminal investigations.
Hello, my name is Sherry McGinnis, and I work with the St. Louis County Police Crime Laboratory. I am one of 2 full-time evidence custodians that take in over 50,000 pieces of evidence every year. We take it all from all of St. Louis County, which does include over 60 police departments, Homeland Security, FBI, DEA, Major Case, just to name a few. When they do bring their evidence, they have to make sure that it is completely documented. Then we put it into our LIMS system, which is a Laboratory Information Management System.
I'm Corey O'Brien, our LIMS administrator. Every item with evidentiary value gets entered into our LIMS system. Our LIMS software was developed by Justice Tracks, a Versatern public safety company, and has been our leading case management tool since January 2017. The benefit of LIMS is that we can electronically track our chain of custody, we can create custom template for analytical reports, and we can deliver clear views of every item being processed within the lab.
After putting all the cases into our LIMS system, then we take it back to our secured vault here in the lab, pending analysis. When it is completed, we then give it back to the submitting agencies. Along other duties that we do give out to all the county hospitals the BAC kits, which is a blood, alcohol, and urine kits. We give out reports to all the officers. Actually, I do believe that we are the face and the gatekeepers of the St. Louis County Police Crime Laboratory.
Hi, I'm Lori and welcome to the Photo Lab. I am the official photographer for St. Louis County Police. I am privileged to photograph many aspects within the department: new employees, promotions, service awards, academy graduation, and officer funerals. I manage the database for all crime scene images and videos, working with the officers, detectives, surrounding municipalities, and the prosecuting attorney's office. I print the images needed for court trials and warrants, while maintaining our commercial-grade printer. While detectives can create their own adult photo lineups, I am the only one authorized to create and preserve all juvenile photo lineups for police departments in and around St. Louis County. I've been in this position for many years now, and I love my job. I love knowing that I am helping with investigations and in court cases.